working in these 2 months at the Marquez-Nostra Laboratory for Cancer Imaging and Therapeutics, and we've been working on ImmunoPET, which is a non-invasive selection tool for patients with lung cancer for antibody therapies. I've been working in many projects at the CIT, but specifically in this few minutes, I will be focusing on zirconium-89 PET imaging of immune response in patients with non-small cell lung cancer. So for this project, we identify one unmet need, which is non-small cell lung cancer. This corresponds to 82% of all the lung cancer diagnoses, in particular of over the 2 million lung cancer diagnoses just in 2020. And current therapeutics standard of care involves antibody therapies, in particular EGFR, so epidermal growth factor receptor targeted drugs. But these develop a resistance after around 1 year. EGFR is responsible for the growth of the tumor. And after around 1 year from the start of the treatment, it will start triggering c-MET, which is another receptor found on the tumor cells. And therefore the treatment won't, c-MET is also responsible for the tumor growth, and therefore the treatment won't be effective anymore. So it has been approved by the FDA for the treatment of non-small cell lung cancer exon 20 mutation, amivantamab, which is a bispecific antibody, which targets both EGFR and c-MET, and therefore can overcome the resistance that comes from the EGFR-targeted monotherapy. Nevertheless, even if the progression-free survival increases, and as well as the overall survival, as well as the time from to the next treatment, the overall response to this drug is 40%. So we need to select accurately the patients that are most likely to respond to the treatment and spare potential non-responders from unnecessary side effects. So current selection methods involve biopsies, which are very invasive methods that are followed by immunohistochemistry, fluorescence in situ hybridization, and gene mutation testing. But being tissue-based analysis, they have different limitations related mainly to the small sampling, which is often not representative of the whole tumor lesion, and also the difficulty to quantify heterogeneity. As you can see here from immunohistochemistry, you can already see how from a small sample we have different positivity to different receptors in the same sample. And so there is a urgent need for a whole body assessment that can select patients. And that's where PET imaging comes into place. So PET imaging works by injecting a radiotracer within a patient. And that radiotracer is a molecule of interest, so an antibody, like in our case, to which it's attached a radioisotope. So this is the radiotracer that decays by emitting positrons, which are the blue particle here. Then they annihilate with electrons that are found within the body of the patient and emits 2 opposing gamma rays. These 2 opposing gamma rays are then detected by the scanner, and through a computer-based analysis, we can come up with such tomographic maps, which have specific regions where the radiotracer accumulates. But before we actually inject the radiotracer within the patient, we have to go through different steps. First of all, we have to produce the radioisotope. In our case, we're using zirconium-89, which through a cyclotron is produced from yttrium-89. Then we prepare the radiotracer. That's done actually at the CIT lab, where we attach to the molecule of interest, so the antibody, the zirconium-89. And then the tracer is actually injected in the patient, which for us is a tumor-bearing mouse. The mouse is then PET and CT imaged through the physics that we were talking before about. So the positrons that are decayed from the radioisotope are then annihilating with the electrons within the body. And through image processing and data analysis, we can get the features from the PET and CT overlap. But we choose zirconium-89. So as we can see here from the zirconium-89 decay scheme, the zirconium-89 has a long half-life of 78.4 hours, which corresponds to 3.3 days. and that matches the biological half-life of antibodies. So it's ideal for ImmunoPET, and that's also because it decays by emitting 22.3% of positrons. Also, thanks to the extended imaging time and the residualizing properties that zirconium-89 have, we can increase the tumor to background ratio, which is the signal to our correspective signal to noise ratio. How to design ImmunoPET agents? So we have the zirconium-89, which is a radiometal. We have a chelator, DFO in our case, and then a targeting vector, which in our case is amivantamab, the drug that the bispecific drug that targets EGFR and c-MET that we're trying to study. Once these get together and the radiotracer is injected within the patient, it will go and bind to the tumor-associated protein targets. So it will go and bind to the tumor, to the proteins that are found, to the receptors that are found on the surface of the tumors, in our case, EGFR and c-MET. More practically, to design, to produce the zirconium-89 DFO, we have to go through a series of reactions for the radiolabeling, which end up resulting in the molecule here, which sees zirconium-89 being chelated by this big molecule, which is DFO, and to which there is a bispecific antibody attached. And then we need to characterize zirconium-89, and that's done through a series of steps for the quality control and the purity of the radio tracer through radio TLC and HPLC. Once the PET tracer is injected in the body, there are two outcomes. Either there is a high uptake at the level of the tumor lesions, and that is the case where the patient is most likely to respond to antibody therapy, and therefore we can proceed with antibody therapy. The case in which there is a low uptake at the level of the tumor lesion will lead to alternative treatments. So our research question is, what is the threshold of responsiveness? What is the level of positivity that patient have to be selected for antibody treatment? So in these 2 months, we've been going through a lot of different experiments. We started with the HCC827 cell culture. HCC827 is a cell line of non-small cell lung cancer. And after around 2 weeks, we injected this cell line on the mice with the subcutaneous injection. And in the next 2 weeks, the mouse will develop a bump on the shoulder, which is our tumor. On day 0, we start the first injection of zirconium-89 and via tail vein injection. After 4 days, which has been shown to be the optimal imaging day for zirconium-89 and for the antibody to flow to the right location, or in the tumor, we perform the PET and CT scan, and this is our baseline scan. After that, we start the AMI therapy. So a group of mice will be treated with the AMI antibody, and another group will be treated with IgG control. So it will be our control untreated group of mice. After the treatment, we start again the PET imaging. So we perform again the tail vein injection of zirconium-89 AMI. And then after 4 days, we perform the PET CT scan, and that will be our post-treatment scan. After the scan, we perform an ELISA with the tumor that have been harvested from the mice that have been dissected. So starting from the baseline CT and PET scan, as we can see here, the uptake of the radio tracer at the level of the tumor, which is where the arrow is pointing at, is very high. So that is in line with what we were expecting, because HCC827 is a cell line of non-small cell lung cancer, which is most likely to respond to the amivantamab treatment. Therefore, it presents the EGFR and c-MET receptors at the level of the tumor cells. So the uptake will be very high at the level of the tumor. So after that, we performed the animal record while we were injecting some of the mice with AMI and some of the mice with the control. As you can see here, the average volume of the control group went really up compared to those treated with AMI, whose growth was inhibited. We can then perform the calculations to analyze how much the tumor growth was inhibited. And we can see how most of the patients had overall 88% tumor growth inhibition. And that's results that are in line again with what we were expecting, as the HCCA27 cell line is most likely to respond to the AMI treatment. By comparing then the baseline to the post-therapy CT and PET scans, we can see again how in the baseline we have a very high uptake at the level of the tumor, whereas in the post-therapy, all the EGFR and c-MET receptors have been degraded and inhibited. Therefore, AMI cannot bind anymore. So not only the size of the tumor decreased, but also the AMI is acting via its main pathway. So it's degrading c-MET and EGFR, and therefore gluconeometinine AMI, the radiotracer, cannot bind at the level of interest anymore. Here we can see these are maximum intensity projections, and these are our CT and PET scans overlap, and we should see in 3D how the tumor not only decreased in size, but again how much the uptake at the level of the tumor decreased. And to quantify our thoughts, we are using the SUVs. So SUVs are activity in tissue over injected activity, and it's normalized by the weight of the animal. And with this, comparing the SUV ratio to the tumor and to the heart. So tumor is the region of interest, and heart is where we have the most, the amount of still available antibodies in the blood flow. So it's kind of our background. And from here, we can see how the SUV ratios from the baseline and to the post-AMI treatment are very much decreased. So that's again in line with what we're thinking. So to validate further this experiment, we would need to perform an ELISA to actually quantify the EGFR and c-MET levels in the tumor that had been harvested. And to validate it further, we will need to try the same experiment, but with different cell lines of non-small cell lung cancer. Just before finishing up, I just wanted to say a few things that I've been learning during these 2 months. So thanks to the interdisciplinarity of the lab, I had the chance of learning more about radiochemistry, biochemistry, a lot more about molecular tumor and cell biology in the cell culture lab, and ultimately a more computational side with image data analysis and PET imaging. I had a chance of working in team in research, which was very fun, other than very educational, and I learned more practical aspects of working in a lab, and ultimately I had a chance of confirming my interest in research and in pursuing academic further education. In the context of the ONAOSI-Yale undergraduate research in advanced instrumentation, I just wanted to show the different instruments that I had a chance of working with through the different projects that we've been working on, and the different software for image and data analysis. Here are the acknowledgements. So thank you, ONAOSI-Yale, for the partnership, and thank you for offering this opportunity to all of us. Thank you very much to Bernadette for welcoming me into her lab, and to the members of the lab, Samantha, my tutor, and Elizabeth and Lindy, that has been working with me a lot, and then all the academic collaborators, and ultimately all the other guys in the program. And here are some pictures of us. Thank you very much.
Simply great. I'm sure that the friends in ONAOSI, they would say that was a good investment. Absolutely. Very, very good. So, any questions?
A lot of instrumentation. Are you happy?
It's wonderful. I loved your learning outcomes in particular.
Thank you.
And partly understanding that you are interested in academic research, actually getting involved in research and seeing that you like it is wonderful to hear that was transformational.
Thank
So
you.
much fun.
She will move to a small university nearby Harvard. But okay, no worries. Yeah, it's a ways.
Evan, you probably have a better.
Better offer?
Better question.
I have a simple question. Your tracer is a bispecific, antibody, so it has two binding sites. Does it for uptake of this tracer, those are plain labeled tracer, does it have to bind to both the c-MET and the EGFR at the same time?
Yes, so we've been performing the experiment also during this timeline, not only with Amy, but also with only c-MET. And we've been seeing kind of the differences between the two, and we're still analyzing, we're still in the process of analyzing how Amy responds better or worse than c-MET alone. So it seems like Amy is most likely to respond better because it overcomes the resistance to EGFR alone, but we would need to also perform the PET scans with anti-EGFR zirconium-89 if we wanted to compare the c-MET and EGFR. And that's, I think, what we're going to do soon with ERLO and anti-c-MET with the carbon-11 and zirconium-89 imaging.
Good,
Thanks.
yes, please.
My question was about the drug Amy. Is it I guess it's probably still in development, or is there like a set time that you give it to the mice that you know in advance, like maybe however long, like 2 weeks, 3 weeks? I can't. You said it, but I can't
so
remember.
we did around the 3 weeks of treatment, and I think that's the kind of the standard here for amivantamab. It's been approved by the FDA only for non-small cell lung cancer mutation of exon 20 in EGFR, which is kind of where they don't have any other options clinically. So they are trying this amivantamab, which it seems to respond very well, but although only in specific patients, which are those that have the EGFR and c-MET on the surface of the tumor cells.
Okay.
And so, yeah, does that answer your question?
Yeah. Thanks.
Good. More? Very good. Thank you once more.